Good morning, everyone. It's my pleasure to have here Dr. Phuong. He is a remote sensing and geospatial scientist, a plant ecologist, and a digital agriculture scientist. He's broadly interested in characterizing plant disturbance interactions using the integration of remote sensing, high throughput plant phenotyping, biochemical and bioinformatics methods, and machine learning. He's also interested in developing novel machine learning image analysis methods and imaging systems to understand these complex interactions across spatial and temporal scales and from controlled environments to field settings. Dr. Dahl is currently an assistant professor of digital agriculture in the Department of Agricultural Biology at Colorado State University. He's also an affiliate faculty in the School of Global Environmental Sustainability and the graduate degree program in Ecology. Dr. Dahl also developed and is currently advising the Agricultural Data Science minor program. Prior to joining CSU, he was a postdoctoral associate at the University of Wisconsin-Madison and the NSF Ascent in Biology Integration Institute. He earned his dual PhD in physical geography and environment studies from the University of Toronto in 2021. So thank you so much for coming. The floor is yours.
Yeah, thank you. Thank you. First of all, I would like to say thank you for hosting me, Dr. Allison Post and Dr. Sigali Emerald for inviting me and hosting my talk. It's very cold today, but the warm welcome make us feel much better. And I'm here today with Dr. Awasila Hassan, one of my postdocs in my group. My name is Phuong Gao. I'm an assistant professor at Colorado State University doing agricultural and ecological remote sensing, especially hyperspectral remote sensing. And today, my talk is from lab to sky, advancing plant sensing and machine learning for climate smart agriculture and ecology. See. Yes. Okay, good. And I just want to provide a little bit more about my background. I'm originally from Vietnam, and I did my undergraduate University of Mining and Geology. So a lot of people wonder why I go from mining and geology to agriculture. But actually, I did my bachelor in surveying engineering, remote sensing, geospatial science. So it's not actually mining, but in the University of Mining and Geology in Vietnam. And then I was a lecturer at the university for 2 years after I completed my undergrad. And then I moved to Taiwan and completed my master in remote sensing science and technology at the National Central University. And then I moved to Toronto, University of Toronto, and I completed my PhD in physical geography and environmental study, specializing in grassland drought and grassland ecology. And then I worked for 2 years as a postdoc at the University of Wisconsin-Madison in a sand biology integration institute that is funded by National Science Foundation. With Dr. Philip Townsend at the University of Wisconsin-Madison, Dr. Peter Wright from University of Michigan, and Dr. Jeanine Cabana-Barros from Harvard University. And I started my faculty job at CSU in August 2023, which has been a year and a half now. So my research lab, Hyper AI Lab, is focusing on characterizing and understanding the mechanism of the interaction between plant and disturbances, including drought, pest and pathogen, weed invasive species, and soil nutrient variation, using the integration of remote sensing, geospatial science, machine learning, biochemical, and bioinformatic methods. And my research spans grassland, cropland, and forest. So my PhD is grassland, my postdoc is forest, and my faculty job is cropland and grassland as well. So, and span from gene to leaf, plant, community, regional, and global scale. Today, I would like to share with you some of the research projects that I've been doing in the past few years by me and also by my members of my research group. And we know that our natural and agricultural system are under great threats of the climate change impact and climate change-driven disturbances impact. For example, drought, pest and pathogen, forest fire, and soil erosion. So it's very important to monitor those disturbances and providing information for protecting our ecosystem to ensure ecosystem services and food security. And before talking about how an ecosystem interacts with environment, we will need to discuss how a single plant interacts with the environment. A single plant is a very complex system with a very complicated functioning and metabolism, including photosynthesis, respiration, growth, and defense, and building a plant structure. And in each of these process, plant functions should play a very important role. For example, chlorophyll, a green pigment that make our plant green to human eyes, play a very important role in absorbing the sunlight for photosynthesis. While non-structural energy, non-structural carbohydrate play an important role in respiration, growth, reproduction, and defense. Right, so understand these variations in shape, we can understand how plants respond to stress and how they perform under environmental changes. And these plant functional traits can be quantified using the field survey, right? We can go to the field and measure a plant property, or we can sample plant tissue to conduct chemical extraction or analysis in the lab to quantify those. However, as you can see that the laboratory analysis and the field survey are very time-consuming and labor-intensive, and sometimes it cannot capture the spatial variation of, in the impact of the disturbances on our vegetation. For example, this area is a grassland site that is affected by drought, and you can clearly see that because of the distribution and the complex composition of different species, the response of different area to the drought impact are not spatially heterogeneous. Right, so the field survey and the laboratory analysis cannot work efficiently in this case. And remote sensing with various platforms, from a lab-based platform to drone, doesn't play video, and satellite platform that provided with valuable information to help us understand the interaction between plant and the services across spatial and temporal scale. And from the lab to the drone to the satellite platform. And what is the fundamental component of remote sensing? As we know that, I mean, you are in Earth Labs, you know that the fundamental component or product of remote sensing is an image. Right? It's like a digital image that you take from your smartphone. This is the RGB image, red, green, blue, so you can clearly see the true color in the real world. And with red, green, blue, you can see the variation in the color between different plants, between different species. But when you look at hyperspectral data with 100 to 1,000 of narrow spectral band, you can see a lot of variation between different species, between different plant, or within the variation within the same plant canopy, right? The variation between the sunlit leaf and shaded leaf. And we know that because we can have the information because we have the hyperspectral sensor that can collect information, reflected information from various contiguous, 100 to 1,000 of contiguous and narrow spectral band. This is an example of the spectra of healthy and stressed plant. In one plot and from one pixel, you can clearly see the differences between healthy and stressed plant, right? Spectra in different spectral regions across the spectrum that reflect the differences in the plant foliar chemistry. For example, the differences in the blue and red region here tell us the differences in chlorophyll content. The differences in this area tell us the difference in yellow pigment, for example, carotenoid or xerocycle pigment. Beyond 1,000, you can see that the spectrum is very sensitive to variation in water, phenolic, lignin, sugar, and nitrogen, and so on. Right, so my lab is focusing on analyzing spectra to quantify zone trend and characterize stress response in different species, in different plant forms, cropland, grassland, and forest ecosystem. And to do that, to quantify the threat from, assess the threat from remote sensing data, we need first the image, right? We can collect the image from the airborne platform, from the satellite platform, and at the same time on the ground, I have a team to collect take the plant measurement, and also we sample plant tissue, and we quantify all of these plant functional traits in the lab. And to do that, we also have to, the image had to be passed through a series of correction and calibration. And you can see that we can quantify a lot of different chemicals in the leaf, like very useful information on remote sensing data. But this is an example of the alternative map of leaf mass per area, chlorophyll, and phenolics produced by Carl Kovac, postdoc in my lab. And you can clearly see, basically, it's the combination of the three colors. So you can see that the area that is more red color, showing that the plant has more phenolic, higher phenolic. The green areas show that plant has more chlorophyll, which means plant is healthy. So for example, this case, in this area, this circular field, this is irrigated field. That's why plant have a lot of water, they perform really well. And they're healthy, that's why they have higher chlorophyll. The area that is just next to it, in grassland area or forest area, that's were not irrigated, right? So they're under drought stress, water stress. So they are not performing well. They don't produce a lot of chlorophyll, but they produce phenolic for them to defend, right? So this map will provide with a very valuable information to help understand how plants interact with the disturbances. And remote sensing can be done at very different scale, from at the lab level, ground level, airborne, and satellite level. And the product can be reflected or images, and we develop various different statistical methods or machine learning methods to extract this information and the information can be used for various different applications in agriculture and ecology. For example, species mapping, plant phenotyping, biodiversity, invasive species, or plant stress, plant breeding, yield estimation, or nutrient management. And to do that, my lab is equipped with, this is the equipment in my lab. We are equipped with several phenotyping sensors that help us to conduct that research project. We have a few spectrometers with more than 1,000 spectral bands that can be used to measure spectra on the leaf or on top of the canopy, plant canopy. We also have a drone, full range drone from 400 to 2,500, 2,500 nanometers. So the drone is $300,000. And we also have a thermal sensor. And especially, I mean, for this sensor, you already know it, so I would not provide you more information. But to analyze the data, all of these systems, we have a server with 32 core and can store, have two storage servers that can store about 300 terabytes of data. And today I would like to provide you with a little bit more information about the lab-based phenotyping system that I just got. This is called Pheno AI Expert. This is a very advanced lab-based automatic plant phenotyping system that has 6 sensors, that have 2 hyperspectral, 2 RGB, 1 3D laser scanner, and 1 fluorescent camera that can capture image of a fluorescent parameter that will help us understand the light efficiency of the photosynthesis. So this is our automatic system that has 2 chambers. One is the palm chamber that can be used to capture, to measure fluorescence on the plant. And it has a dark adaptation chamber as well. To measure that, you need to put the plant in the dark adaptation state for about like 20 or 30 minutes. And it has a dark adaptation chamber as well. And it's an optical chamber where we have hyperspectral RGB and 3D laser scanner. And you can see it has a conveyor, so we can move the sample inside and outside of the system. And this is a side view of the system. And you can see that it has a robotic arm to move the sensor up and down. So it's very flexible and work with a very large plant of 1 to 1.5 meter. It can be moved very close to sample to capture images of the root and seed. So we can do root phenotyping, seed and soil phenotyping as well. And it has 1 RGB camera on the side to capture images from the side. And the sample can be rotated 360 degrees. So it can provide a 3D view of the plant. This is the 3D view of the plant. An example of some data that we collected. So they provided a very precise and high resolution data to help us characterize and understand the mechanism of the interaction between plant and disturbances. And you can grow plant in greenhouse and apply various different treatment, drought treatment, temperature treatment, disease treatment, and understand and use that system to provide us with a complete picture of how our plant responds. And this system will be installed in the next few months. And today I would like to use some of the research projects that I came up with in the past few years using lab-based phenotyping and airborne phenotyping in characterizing the interaction between plant and stressors, especially the interaction between plant and insect herbivore, plant and drought, and how we use imaging spectroscopy and plant phenotyping in precision agriculture. The first project is characterizing the impact of insect herbivore on our aspen forest. So this study focused on spongy moth. This is an invasive species that was introduced in 1869, and it's one of the most destructive species in the US, and it feeds on about 500 different species. And this is, you can see how it impacted. It was introduced to the eastern GS in the eastern region, and you can see that it's quickly spread out to many different states in the eastern and the Midwest, and across various different host plant species. So it's very important to understand how these species interact with our host plant for providing information for conservation. And this is a photo of the field that I studied on your right-hand side. You can clearly see the variation in the defoliation between the three plants that are next to each other. They're planted next to each other, but they're different genotype. The same aspen forest, but different genotype. You can clearly see that one plant was completely defoliated. One was defoliated to some extent. One was barely defoliated. They survived. They can survive. So we assume that there's a genetic variation in how plants respond to the impact of insect herbivore. So we conducted the study in this common garden with 1,825 different genotypes, a tree of about 520 different genotypes. And the objective of this study is to characterize the genetic variation in plant chemical defense and how plants allocate their resources when they impacted to survive, and identify the gene or genomic region that is involved in the response process. And I integrate airborne remote sensing with airborne hyperspectral and LiDAR and multispectral remote sensing with biochemical and bioinformatic methods and biological modeling. So we model the insect, different instar of the insect and how they impact the plant in different instar. And this study focuses on two main defensive traits, including phenolic glycoside and condensanin, that play a very important role in plant chemical defense, especially in aspen. Phenolic glycoside confer toxin to herbivore and reduce their digestion, while condensanin prevent the plant from the oxidative damage and inhibit microbial functioning for plant to survive and alter nutrient cycling. And we conducted a series of experiments and few data collection campaign, including ground data collection campaign. So we use cold pruner to collect the leaf. We cut some branches, and every week, and every time we fly our sensor, and we fly aircraft and drone hyperspectral sensor, and we also have a LiDAR sensor called VTOL system, vertical takeoff and landing system, and we collected the data throughout the growing season. This example of hyperspectral images in false color composites of the garden before, during, and after defoliation. So the plant was impacted from early June to late June. And then the moth pupae, and they stop eating, and the leaf grow back in early July. And you can, after the leaf grow back, you can clearly see the variation in different color, right? Representing the changes in plant chemical, in leaf plant chemical. So today I would like to understand how those chemicals change under the impact. And we collected the leaf sample, we free dry them in a freezing condition to make sure that the chemical is not degraded. And we grind them and measure a dry spectra of those samples. And we send out those samples to quantify nitrogen, carbon, phenolic glycoside, and condensanin, and non-structural carbohydrate, and some other parameter. We also measure the spectra of the fresh leaf when we cut them down from the tree, and we also fly airborne sensor to collect images. So these are the spectra of the data set at the 3 level. And then we use a chemometric method, for example, least square, partial least square regression to link spectra that we collected in the field in the lab with the chemical content that we quantify in the lab, and we build the model. Basically, we identify the spectral region that is sensitive to variation of each chemical. For example, a variation in condensanin is very sensitive to the spectra at about 2140 nanometer, right? And the variation in phenolic glycoside is very sensitive to the variation in spectra at about 1670 nanometer. And these these absorption features, you can see that are preserved from different scale. from lab scale to the image scale. So it's very reliable. And this the absorption feature has been reported from previous study. And this is the result that we get. So if we use dry spectrum, we have an R-square of 0.9, 0.98. And not only from my study, but from a previous study from a colleague in my lab, we showed this exactly the same study. And this means that in the future, if you give me that sample, you don't need to do chemical extraction. I can tell you how much phenolglycoside, how much conductance we have in that leaf. And the accuracy decreased for the fresh leaf model and airborne model because of the water absorption. Water is very abundant in the leaf, right? And water absorbs the signal from 1,000 to about 2,500. That's overlap with the absorption feature of those chemicals. But I think the result is still promising. Taking into account the fact that we measured from distance. And then I built the model and I applied prediction and I quantified it and produced the trait map. This is the map of phenolglycoside and condensed channel. With that, we can clearly see the spatial and temporal variation in those chemicals before, during, and after the defoliation. And you can clearly see that condensed channel increased significantly under the impact. And then after the impact, when the leaf reflects, condensed channel decreased. So, I mean, condensed channel is very responsive when they impacted, right? But it's not the legacy effects. And you can see phenolglycoside, on the other hand, in contrast, remain quite stable when they're impacted, the plants were impacted, but increased significantly after the defoliation. So this is the defense mechanism of the two chemicals. So it means that plant used condensed channel defense mechanism when they impacted more than phenolglycoside. But after the impact, plant may remember, oh, I was impacted before. I need to increase phenolglycoside now to prepare for the next target, for the next attack. And you can clearly see the variation between very contrast pattern in the variation between the two chemicals. And people will question, like, how can you quantify this shade from distance, right, from a sensor on an aircraft? This is the comparison between the lab work and the image. The result from the chemical extraction in the lab and from the image. You can see that there's some differences between the two, but the pattern is very similar, right? The variation pattern, condensate increase, decrease, and at the bottom, condensate increase and nitrogen always decrease under the impact and after the impact. So this shows the utility of hyperspectral data in quantifying those chemicals and their variation under different disturbances. And we also analyze the intervariation between primary metabolite and secondary metabolite. You know that primary metabolite play important role in plant growth. Secondary metabolite play important role in plant defense. So we want to see how they vary under the impact and how plant reallocate their resources. And you can see that before the impact, nitrogen is quite high, right? Condensate is high, but not super high. But under the impact, you can see on the right-hand side there, condensate increased significantly and nitrogen decreased significantly with the impact. Because at that point, plant may not need a lot of photosynthetic nitrogen for photosynthesis. They stop photosynthesis or they reduce, down-regulate the photosynthesis to defend, invest more resources to defend. That's why they produce more dentate and phenolglycoside. And after the defoliation, you can see that the point cloud moves toward phenolglycoside. This means phenolglycoside is more of the legacy effect. This provided with valuable information to understand the mechanism of plant response to insect overall, right? And there are also genotypic variation as well. And of those chemicals, and you can see that some genotype respond much better than other. For example, this one, about 620, 52, that's the genotype of this one, while 206 genotype respond much better. So these are the plants that survive during the impact. So this helps us to understand which genotype respond better. They provide information for conservation and also provide information for plant breeding as well in the future. And in terms of plant breeding, we also did bioinformatic analysis. And usually we link trait with genomic data to identify genetic variant that is involved in the variation of the trait. And in this study, we link spectral with genomic data. So basically, and genomic is sequencing data using genome-wide association study. So basically, we identify spectral band or spectral region that is sensitive to variation in the trait. and link those bands with the genomic data. For example, this is a demonstration of how to link genomic data with band 1660 that is sensitive to variation in phenol glycoside. And you can clearly see that we can find some genomic variant or a genetic variant that is involved in the chemical response. We provided with the information for plant breeding. The second study I would like to share is the drought impact on native and invasive species. That is one of part of my PhD research I did in Toronto. And as we know that when plants healthy, the leaf of plant are green, and they have a lot of chlorophyll content in the leaf to play a very important role in absorbing the sunlight for photosynthesis, right? And most of when the plant is healthy, most of the absorbed light will be used in the photochemistry, right? Photochemical reaction of the phase of the photosynthesis. But when they're impacted by drought, the photosynthesis is down regulated. So plant don't need a lot of energy at that point, right? And plant are very smart. They reduce chlorophyll content. That's why when you see plants affected by drought, the leaf turns yellow or brown. And then they convert these event more resources to increase yellow pigment, for example, carotenoid or xanthine cycle pigment to help plant convert excess light to heat and release back to atmosphere to protect themselves from oxidative damage, right? So photochemistry decrease and heat increase. So my PhD research focused on characterizing how native and invasive species respond to drought through analyzing the variation in plant functional trait and light efficiency. And we compare how native and invasive species respond, whether invasive species is more tolerant to drought or native species is more tolerant to drought, and why. And I conducted a series of experiments from research greenhouse to rain exclusion shelter and natural drought. So I collected plant sample and grew them in the greenhouse and applied different water treatment from 100%, 75, 50, and 25% of field capacity or full rainfall and examine how plants have different water treatment respond. And in the field, I built about 10 rain exclusion shelter with the greenhouse plastic that block the rainfall but allow sunlight to penetrate and compare the plant inside and outside the shelter. And we also have the natural drought. We'll see whether our experiment can be translated to the natural drought. Right. We compare the data from the wet year in 2017 with the drought year in 2016. And I worked with one of my colleagues to develop an affordable plant phenotyping system. It's not like the one that I introduced you. The one that I just introduced $500,000. But this one is about $12,000. We have a hyperspectral camera, which is about $80,000. And then we develop a frame, which is about $12,000. It has a moving platform at the bottom, we call linear state, to move the sample. And we are simulating the F1 fly. And we move the system to put the sample on the linear state and move the sample. And the sensors on the top. So it's a very cool project. And then in the field, I use helicopter with the hyperspectral sensor. And we cut a small hole at the bottom of the helicopter, and we put the sensor outside of that hole. Images. It's fun to fly a helicopter and capture images. And then we also, part of this study is to characterize how topographic variability affect or control our plant response. You know that topography control distribution, composition, and how we respond to environmental stress, right? In this study, I characterized the topography into 6 different categories and examine how plants respond to drought in each different category. And we did develop various motion and different methods, especially object-based image analysis. I don't have the time to explain object-based image analysis, but it's basically when you analyze remote sensing data, you can analyze every single pixel. But in my method, I group them into to form different objects. I group pixel together, pixel with a similar spectral properties together to form image object. For example, all the pixels of your building will be grouped together. And it's a Earth lab, and I only analyze that object instead of analyzing all the pixels inside. So it's called object-based image analysis. And we produce, the model has achieved an accuracy of almost 98% when we detect early drought after 3 days of the water treatment. And we also use machine learning to map the distribution and composition of native and invasive species in the field. And then we compare that distribution with the topography map. Now, what pattern you can see here? You can see that smooth brome, an invasive species, occupy the dry area, high elevation, steep slope, and dry area. That is less favorable to native species, right? And so that makes them more tolerant to drought and thrive in a water-limited environment. In contrast, most of the invasive native species are growing in the flat and lowland area. So this is the mechanism of the invasion of this species. Smooth brome is migrated from Asia, and they're more tolerant to drought. They grow in a hot environment. So the mechanism is that, or strategy is that they occupy the disturbed area that is less favorable to native species. And in terms of topography, so I produced a chlorophyll map of 2017, the wet year, and 2016, the dry year, and compare the rate of change between the 2 years. And you can clearly see that the impact of drought is not spatially homogeneous, right? Some areas greener, some areas is more yellow. So greener means that plant is healthy in that region. And I want to compare that with the species distribution map. And you'll see that this area is actually smooth brome. So smooth brome, they don't only occupy the dry area, disturbed area, but they're also more tolerant to drought. When the drought happens, they're more tolerant. You can see that the chlorophyll remain much more stable than other species in other areas. So this is the mechanism, because they are adapt to the dry environment already. So when it's drier, they're more tolerant. They withstand that drought impact. So this provided with information to help us understand the mechanism of the invasion, right, to help protect our native ecosystem. And we also do within and between species trait variation. You can see that smooth brome, the trait is much more stable than all other native species. However, within smooth brome itself, if they grow in different environments. So smooth brome that grow in the dry environment, they're more tolerant to drought, and they're much more, how they adapt to the environment, they're much more tolerant to drought when the drought happens, compared to the same plant growing in the wet area. You can clearly see the distinction, right? If they grow in the dry area, they'll be healthier than the same species that grow in the wet area when they're affected by drought. And then we later on we scale up that analysis to study California drought in a larger scale in 2015 and 2016, and we analyzed the intervariation between the in the plant function trait. And when you can see that before drought, that the point cloud nicely distributed in the center of the trait space, but when they're impacted by drought, the trait space shift toward non-structural carbohydrate and phenolic. Right? They need to invest more resources to phenolic for them to defend when they're impacted by drought to survive. And then when the drought went away, you can see that trait space slowly recover, right? They move back to the center. This very useful information. And with that number of data sets, lab data set that we have been collecting, we collaborate with researcher from Germany to build a global trait model, transfer model that can be used for different species and different bio. And this aligns really well with the goal and vision of the NSF-funded Ascend Biology Integration Institute that I was part of. And the last one is the last research theme I would like to introduce is how to use remote sensing and plant phenotyping in precision agriculture in my lab I'm doing at Colorado State University. And the first study is develop a method for quantifying or for detecting early infection in sugar beet under the impact of soil-borne disease, and develop a computer program to automatically detect and characterize the impact and identify the disease-resistant traits for plant breeding. And see that soil-borne disease is not easy to detect because they affect the sugar beet root first. And they don't show the symptom in the leaf. Leaf are still green. You can't see it. You might see it from the hyperspectral sensor that can see the band in above 1,000 nanometer, but from a human eye, you cannot see it. And you can see it at the later stages when it's already very severe. But early detection very challenging in soil-borne disease. And then we conducted a series of experiments from research greenhouse to field experiment. So we grow plant in the greenhouse and inoculate the disease in the soil and examine how the impact. And we use the same gene, we grow the same genotype in the field, and we use the drone hyperspectrum to characterize that interaction. And this is done by Dr. Hassan here. We show that the sensor can effectively detect early infection in the roots, and it can easily distinguish between healthy and infected plant. And it can also characterize different severity level, whether the plant is healthy or it's stunted or it's withering or it's dead, providing farmer with information to act like how to treat their disease when it's impacted. And we use a statistical and machine learning method to identify the spectral region that is sensitive to the disease. When disease happens, which spectral region will be responsive to that. But one of the most important information that a farmer needs is what sort of disease my crop is affected by, right? Whether it's rhizoctonic disease or fusarium disease. So I can use the right chemical treatment. I can apply the right chemical treatment. So in this study, we also analyze the utility of our spectral data to distinguish different types of disease. And you can see that we have two diseases, fusarium and rhizoctonia. And you can clearly see that the spectral response is different in different spectral regions, especially at about 400 nanometers. This was about 560 and about 650 and 700. We are very useful for distinguishing different types of disease. And then we can also quantify the area of the leaf, right? The percentage of the plant that is affected by the disease. And in the field, you can see that we can, not only in the lab, but in the field, we can tell whether a plant is healthy or infected, and the probability of the infection as well. I say this plant is infected, but what is the chance of that plant being infected? 50%, 80% of this area is infected or not. Not only the hard classification whether it's infected or not. If infected, we also tell farmer that, oh, it's a 50% chance that this plot infected. And then we are analyzing plant foliar chemistry data to quantify pigment, for example, chlorophyll, carotenoid, and non-structural carbohydrate, nitrogen, and phenolic to help better understand the mechanism of the chemical interaction. And we also link with the genomic data to help identify the important trait, disease-resistant trait for our plant breeding. And this study was conducted in RDAC in Fort Collins. And this year we'll expand our analysis, our experiment to incorporate 100 different genotypes of sugar beet in a sugar beet field and experiment field in Long Lake, Colorado. And the last study is about the interaction between crop and soil, nutrient runoff and soil water runoff. So this study was conducted in Canada, and also now it's extended to my new study in Fort Collins at Colorado State University. This study characterized how plants perform under the impact of nitrogen runoff and water runoff. So we have thousands of different treatments. Right? We apply nitrogen treatment, crop rotation, or the combination of different treatment, and we use the integration of hyperspectral, thermal, and multispectral data from at the ground level and at the airborne scale to characterize that plant performance. And we went to the field and we also collected a lot of soil sample, and then we dry them and we measure spectra, and we quantify the soil organic carbon variation in the soil in different plots. And then we want to analyze how the change variation in the soil water and soil organic carbon variation will impact our crop health. And we can use spectra to quantify soil property. I'm not sure if you heard about using imaging spectroscopy to quantify soil quality before, but from our study, we can show that it is possible. Right, so we have an R square of about 0.6, 0.7 using spectral data to quantify soil organic carbon. And then we develop machine learning model to classify whether a plant is healthy or is under nutrients or under stress. And if it's under stress, it is under nutrient stress or it's under drought stress. Similar to distinguish different types of disease, right? And we have different spectral region that is sensitive to disease, drought, or nutrient deficiency. And this is, we also produce a trade map of the entire field. And you can clearly see that in the nitrogen treatment, the green plot here shows that plant is very healthy. And the other plot that when the color change to yellow, that plant is less healthy. They're experiencing net nitrogen deficiency, but at different level of severity. So when you tell farmer to apply nitrogen fertilization, you tell them don't apply the same rate, right? Don't just apply the same 10 kilograms for each plot, but apply variable rate for each other plot. And in this field, you can see a very interesting pattern here, right? This area, that's the area where plant is experiencing drought. So we install a lot of pipes under the ground to drain out water faster. You generate drought. You don't see it from above ground, but from the image, you can see that sort of interesting pattern. And now we expand that study and we incorporate plant phenotyping with root phenotyping. So we pull up the root and we take 3D images of the root system to characterize how plant respond to drought and nutrient deficiency above and below ground. And one of the problem that we are tackling is in remote sensing, people do it separately. People do it in the lab separately, in the field separately, and they develop the model separately. However, the pattern that you observe in the lab may not be translated successfully to the field because of the atmospheric effect in remote sensing, right? Or of the impact of a lot of other parameters to our plant. So the conclusion that we make in the lab may not be the same field. What my lab is trying to do, and Dr. Hassan just complete analysis, and the paper will be submitted soon, is how to extract the information from different scales and combine them together. How to extract the information from the lab model and field model on the ground that has high accuracy and feed it into the image model to improve our prediction and trait quantification. And that study will be submitted soon. And when we incorporate information, we see that accuracy increase about 10%, 15% most of the time. And then you'll see that plant phenotyping on the ground may not be able to provide you with a complete picture of how plants respond. So in this study, we expanded to incorporate the plant sensor with the soil sensor. So we use soil sensor, moisture sensor, and soil pH sensor to collect all the information and incorporate with the plant sensor above the ground to improve our prediction. And the multimodal AI is very important. How to incorporate different sensors, by how to combine image with the soil sensor, with soil moisture variable is also a question. And another new research area is how to combine computer vision model, the model that help analyze the image with the language model that incorporate the feature from description from text. This is very important, improve the prediction accuracy and reliability. For example, when you analyze the image and you say that you apply the prediction model, you say that this area is affected, this plot is affected by fusarium disease. But actually, from my soil measurement of pH, I say that soil pH of this plot is 6 to 8. That is not favorable to fusarium. Fusarium is only emerge and thrive with a pH of about 5 to 5.50 or 5.5. So incorporating those descriptions will help us improve our accuracy and reliability of the prediction model. That's what we are pursuing in my lab. And when the method has been developed and verified, we can apply it to many different disturbances and different crop and disturbances in area, especially in the state of Colorado. With that, I would like to stop here and thank you for listening, and I'm happy to answer questions. Yep.
Yeah, so this may just be because I'm not that familiar with invasive species, but I found that result about the like spatial mechanism
Yep.
for the invasion really interesting, because I think a lot of times when I've thought about it, it's been more of like a time thing, like maybe there's some disturbance and the invasion comes in. But if it's that they're like invading in like locations where the native plants aren't doing so well anyway, that suggests a totally different approach. So I'm curious if there's any kinds of applications to restorations or combating native species that you think comes out of research.
Yeah, that's a very interesting question. And I mean, occupying or invading the disturbed area that is not favorable to native species, one of the mechanisms of the invasive species. So with that, we will know that if we know which species is suitable to grow in which different region, topography region, we can provide the conservationist with very valuable information how to control it. So I know that, okay, this species cannot be grown in this area, so I don't need to pay attention to it. I don't need to develop a program to control it, right? And I know that it's gonna be invading that area. So I better plan to sort of like control it in that area. That's of course spatially, but it's depending on the, let's say, the management program and of the state or of the country. And sometimes we don't need to control them because we might need them to grow in that area to, for example, for livestock grazing. But smooth bone is an invasive species in my area. But smooth bone is also for livestock grazing as well, right? So we know that if we grow in that area, it can survive or not. And whether that area is the best for growing in this area, and whether we can raise cattle or cow in that. So it's a complicated question, but yeah. Yep.
Thank you for your talk. It was all very interesting. I was wondering for I think the first section you showed on sort of mapping differences in traits with the moth, the defoliation. Yep. When you do that for training the spectral trait model, is that a single point in time that the data comes from? And then do you think there's any considerations for like how transferable that model is throughout the growing season to different points?
Yeah, that's a good question. And actually, due to the variation in the chemical, so usually we don't only use the data from one date at one time to build the model. So what we do is that we don't sample all of them. We have 1,800 trees in the field, in the garden. We don't sample all of them. But we sample. We have historical data before, and then we do a clustering analysis. We cluster data to about 20 different clusters, and we randomly sample based on the percentage of the number of trees in each plot, in each cluster, and we sample about 80 trees every time. And we sample plant foliar tissue across the growing season, right? In May before the garden was impacted, and in June, in July, and in August, September. So make sure that our model is more representative. And if you only have the low content in early growing season, and later on in the later growing season, it increased significantly, you can't use that to predict, right? So, but I will say that it's representative, and it can be used for different images of different year as well. But I would not say that this model can be transferred to different species. The different species have different chemical content, leaf structure, leaf hair, right? So those will control the spectral response. So I would not say that it can be transferred to species, but for this species specifically transferable. And we already, when we train the model, we only use about 70 or 80% of the sample, and then we use independent sample to validate. Yep.
You say that you also work with grasses a lot.
Yep.
Have you been able to detect different grass species using hyperspectral like remote sensing?
Yep.
And mainly through lab work or also, for instance, through satellite or drone type?
I use for that study for that grassland study, I use integration of field data and the airborne images, not the lab work. So in the field, we were after when we flew our aircraft to collect images, we also go to the field and measure. Or we collect the GPS coordinate of the species in that area. The area. For example, this area I circle it 10 by 10 square meter, or 10 by 10 meter is smooth road. That area is goldenrod. That area is the orchard crab. And then I link with the those data with the airborne data as a training sample to train the model. And then I successfully mapped the distribution and composition of the species. And you can see that we have an accuracy, achieve an accuracy about 90% in the species mapping. Yeah, about 90% accuracy. You can map the location of different species.
And do you foresee a future that at some point that might be possible with satellites as well? Or do you think
Yeah,
that's
people use satellite all the time. And Dr. Sigali Amro also know that we use satellite data.
so even for grass species?
Recipe is challenging, depending on how complicated your species composition is. For mine, it might not be possible because it's in a very small area, but has about 6 or 7 different species, and the size of the plant is only like 20 centimeter. So the image that I use is about 10 to 20 centimeter, with about 300 different channels. The spatial resolution is about 20 centimeter. Because my grass species is very small and highly mixed, but I'm not sure. Maybe you can do it in the grid, in the central plane where species quite homogeneous. And we actually extended our study to central grid plane using the satellite data, but not to classify different species, but to characterize the dry response of species. And I don't have time to explain that, but we found the opposite trend. So my PhD found that plants grow in the dry area, adapt to the drought, and when the drought happens, they're more tolerant. But and that is also the same finding by my collaborator, Dr. Allen.
Not to say. I
However,
love that paper, yeah.
the student, the master student that Sydney Hadberg, she just graduated in June, and I co-survived with him, found the opposite.
Really?
Yeah.
Interesting.
Actually, that is reasonable, and it's not like I would say there are 2 conclusions have been made. One is that when plant grow in the dry areas, more tolerant to drought, and when drought happens, they're more tolerant. They're adapted to that. But the other will, a lot of other people make the opposite conclusion, and that in that Sydney Hadberg paper, which was submitted a few months ago, we discussed that. So 2 sources have found the opposite conclusion compared to our master student. But yeah, in the literature, in previous study, there are 2 conclusions. So what we're trying to do is try and understand why is that. Is that because of my studies, more fine scale, it's in just 1 kilometer, 2 kilometer by 1 kilometer area, while Sydney studies cover the entire central plain. But that's an interesting thing that we find. But I don't have time to explain that for today. But you can later on you can read that paper in my paper to see 2 opposite conclusions, 2 contrast conclusions in pattern.
Very interesting.
Yeah. And we understand what is the sort of the factor that control that. It might be because of the microclimate, the small scale, the control of the small scale topographic variability, or soil type or soil nutrient level of different field, and whether that sort of the impact of that factor or the influence of those factors can be translated in across different scale, right, from fine scale to large scale. But we did find very interesting pattern to contrast pattern. Yep.
I have a question kind of related to that, where I guess I'm just wondering when you are identifying grass species based on their hyperspectral signatures. Do you ever find that their ability to regulate their chemical traits confounds your ability to classify which species they are? Like, can they kind of functionally look like others drought tolerant species when they are upregulating their chemical traits to improve their drought resilience?
Yeah, I think you can do that. And in that, you have spectral data, you can quantify. You cannot quantify fluorescent directly, but you can use, for example, photochemical reflectance index as a proxy of the light UV efficiency in PRI as an index. And you can quantify a lot of traits, for example, pigment, especially xanthine cycle pigment, and carotenoid that play a very important role in helping plants to defend themselves from oxidative damage. So that's intervariation or conversion between chlorophyll and carotenoid, right? When the drought happens, chlorophyll decrease and then carotenoid increase. So plant reallocate resources. And nitrogen, photosynthetic nitrogen decrease by the impact. And the phenolic and non-structural carbohydrate will increase. That's my speculation in drought. I did not do non-structural carbohydrate in my PhD, but from other studies who show that the phenolic will increase, anthocyanin will increase, carotenoid will increase when the drought, but nitrogen will decrease, leaf water will decrease, and also pigment will, like chlorophyll will decrease. So we can, from that, you can help better understand how plants reallocate their resources to what defend or to grow when they're impacted or when they're healthy. And for species classification, you can also use trait as well. And people use trait data to classify different species because different species will have different trait composition. Even when all of them are healthy, they have different trait composition. That as well.
This is also kind of related, but I'm just curious if you could do like a quick, what is the future of hyperspectral, particularly from the satellite perspective?
Yep.
A lot of your studies are on very fine scales. And so part of the question was like limitations in spatial resolution, temporal resolution, all those things. But there are some hyperspectral missions coming out soon. There's SGB. I just wonder if you like, what's your take on where that's headed over the next like 5-10 years?
Yeah, in 5-10 years, it's going to be a booming of hyperspectral data, and especially SPG. Dr. Emerald also know that. I think in the past few years, in the past, there were not a lot of hyperspectral satellite sensor like Hyperion, right? But not a lot. We have NMAP. I was launched. I'm not sure it's working now. That was an issue a few months ago. But the NMAP, Prisma from Italy, SVG from Surface Biology and Geology from NASA Jet Propulsion Lab, and the system will be going to be launched in the next 3 years, in 2027. And the data is free. The spatial resolution is 30 meters, the same as Landsat. 16-day, if I remember correctly, 16-day temporal resolution. Provide free data sets for you to upscale this to a regional and global scale. But it's going to be a huge data set that you have to be prepared and how to optimize our system to be able to analyze that data. And we have Canada. Canada was launched a few months ago, and they just have a first few data set released a few days ago when I check it. And they also have some webinar of using that. They also have emit sensors on the International Space Station. So, but I think we have a lot of opportunity in the future, and people are collecting the ground data all the time. So if you can build an open source platform for all people to contribute ground data to link with the satellite data, that will be very valuable in the future, in the next 10 years. And actually, I was part of the CalVal program. I'm not sure, Sibeli, you are in that CalVal program, SHIP campaign?
I have been on algorithmics.
Yeah, so we have, and the paper was accepted a few months ago, and it's going to be released in the next few weeks. Maybe this week or next week. That's a SHIP campaign that is a calibration and validation campaign of that satellite. And we conducted data collection, few data set campaign in California for a few months. And they published several paper about that campaign. There's going to be big opportunity in. But now the spectrum is not, it's not available at a lot of different scale. I can do it because I have a drone or I have an aircraft, and a lot of people don't have access to that. But in the next few years, when SBG is launched, then people may not do Landsat anymore. They'll do SBG data. I provided a lot of information to better understand how our planet ecosystem interact with environmental change at the user global scale.
I think we're at time. Thank you so much. That was
Yeah, thank you.
I think Sabelli and her team's gonna have lunch with you, and anyone else who wants to join, they'll be down in the cafeteria, so.
Yeah, I'm happy to chat more about. And one of the good opportunities in integrating lab-based phenotyping with remote sensing data. Remote sensing data, sometimes we do prediction, we do classification, we quantify trade, but you know that the accuracy vary